The next organelle that we're going to be talking about is the endoplasmic reticulum. So the endoplasmic reticulum is another membrane-bound organelle, and for that reason it's only found in eukaryotic cells. Bacterial cells do not possess this structure. So the endoplasmic reticulum, it extends directly off the nucleus. So if we have the nucleus here, which houses the genetic material, extending directly off of that, we have the endoplasmic reticulum. So there's two different types of endoplasmic reticulum. There's the rough ER, or the rough endoplasmic reticulum that we see here in the pink, and then extending off of that in the orange, we have the smooth ER, or the smooth endoplasmic reticulum. Now, collectively, the endoplasmic reticulum is involved in transport and storage. Now, the two different types are involved in transport and storage of different things, but the collective function is transport and storage. So looking at the rough endoplasmic reticulum, or the rough ER first, we can see that it is studded here on the outside. It is studded with ribosomes. So there are ribosomes attached all along the surface of the rough ER, which gives it a rough texture, which is why it's called the rough ER. Ribosomes, we know, are associated with protein synthesis. That makes the rough endoplasmic reticulum indirectly associated with the storage and transport of proteins. So typically, proteins are produced on the ribosomes that are attached to the rough ER. Those proteins are then stored in the rough endoplasmic reticulum and then eventually transported out of the rough endoplasmic reticulum to another location. The smooth endoplasmic reticulum down here is not studded with ribosomes. It has a smooth texture. It is associated with transport and storage of non-protein molecules since it lacks ribosomes. So it's going to be typically involved with the storage and transport of things such as lipids, or non-protein molecules, of which lipids is a non-protein molecule. So collectively, endoplasmic reticulum is associated with storage and transport. The rough ER is involved in the storage and transport of proteins, whereas the smooth ER is involved in the storage and transport of non-protein molecules, primarily lipids. Another membrane-bound organelle present only in eukaryotic cells is the Golgi apparatus, or the Golgi body. The Golgi apparatus is responsible for protein modification and shipping to kind of the final location within the cell for the protein, which may be the cell membrane or potentially even the lysosomes if we're talking about digestive enzymes. So what the Golgi apparatus is going to do is it's going to receive often proteins that have been sent to it by the rough endoplasmic reticulum. So here again we have the nucleus in the center. Extending off of that nucleus, we have the rough ER, and we can tell that it's the rough ER because it's studded with all the ribosomes. And then over here we can see a little bit of the smooth ER since we don't see those ribosomes. But looking at the rough ER, we can see that the ribosomes are on the rough ER are producing proteins, which are then being stored in the rough ER and then transported out of the rough ER to the Golgi apparatus, where they are chemically modified and then sent to the final location, where it looks like the final location, for this specific example, is to be exocytosed out of the cell. But these proteins could also be sent to the cell membrane to be embedded for transport, or again, potentially the lysosomes to serve the role of digestive enzymes. So another thing that we have to add to this is transport vesicles. So how is it that the proteins are able to get from the rough ER to the Golgi apparatus, and then from there to the cell membrane or outside the cell? So there are two different types of transport vesicles that are used within the cell: transitional vesicles and condensing vesicles. So transitional vesicles are going to run from the endoplasmic reticulum to the Golgi apparatus. So when the proteins are stored and then transported out of the endoplasmic reticulum, the rough endoplasmic reticulum, they're going to be transported in transitional vesicles until they get to the Golgi apparatus. Then they're going to be chemically modified in some way, maybe by adding sugar groups. And then they're going to be repackaged in new vesicles called condensing vesicles. And the condensing vesicles will carry the proteins to their final location within the cell, which, like I said, in this case is going to be outside of the cell via exocytosis, but it could be the cell membrane, it could be to a lysosome. So knowing these different types of organelles that we just talked about, we can kind of see this nature's assembly line that's created. And it all starts back to the DNA. DNA is very important. It is housed in the nucleus, and it essentially serves as the instruction set or the recipe for how to build all the various proteins that can be built in a cell. Functionally, DNA doesn't do anything. It just really, again, serves as the instruction set. What it tells the cell how to do is produce proteins that can functionally engage different processes within the cell. So in the nucleus, we have the DNA that is turned into RNA at specific gene regions using a process known as transcription, which we'll talk about when we get to the microbial genetics lesson. And then in transcription, the messenger RNA that we produce is able to then, or after transcription, the messenger RNA that we produce is able to link up with a ribosome to engage in protein synthesis, which we know as translation. So if that ribosome is found on the rough endoplasmic reticulum and that protein is produced and then stored in the rough ER, it can be transported out of the rough ER in those transitional vesicles to the Golgi apparatus, where it can be chemically modified and then eventually sent to its final location within the cell, such as the cell membrane on those condensing vesicles. So it kind of forms this assembly line starting with DNA in the nucleus, all the way down to turning it into RNA and joining with the ribosome and going through the endoplasmic reticulum and the transitional vesicles and the Golgi apparatus and the condensing vesicles. Two other structures, these two kind of work hand in hand, vacuoles and lysosomes. Both of these structures are membrane-bound organelles, so both of these structures are found in eukaryotic cells only. These two structures kind of act almost like the digestive system in a cell. Vacuoles, they serve the role as storage components. So if a cell would engulf nutrients, those nutrients would be temporarily stored inside of a vacuole. Or if a phagocytic cell, like a neutrophil, engulfed a bacterium, it would temporarily store that bacterium inside of a vacuole. So a vacuole is just a membrane-bound compartment that essentially stores things while they're awaiting digestion. So it's just a temporary storage compartment. Eventually, a lysosome will fuse with the vacuole, which is what we're seeing here. Lysosomes are what actually contain the digestive enzymes that are needed to break down nutrients or potentially destroy pathogens that have been engulfed through phagocytosis. So lysosomes will always fuse with the vacuoles, and the lysosome will release the digestive enzymes into the vacuole and break down whatever the vacuole was storing, whether it was storing nutrients or food in the form of that will then be processed into nutrients, or whether it was storing a bacteria that needs to be engulfed and destroyed if it was a phagocytic cell. But vacuoles are merely just storage compartments while awaiting digestion. Digestive enzymes will come from the lysosome to then break down whatever was being stored in the vacuole. The only thing that you really need to know with mitochondria for eukaryotic cells is that, again, they are the powerhouse of the cell. I'm sure you've been hearing this for quite some time. So that the mitochondria is where ATP is generated. I think most of us already knew that. One thing I do want you to remember, though, only eukaryotic cells have mitochondria. Prokaryotic cells, such as bacteria, do not have mitochondria. They obviously need to produce ATP, though, or energy, So prokaryotic cells will produce their energy using the cell membrane, whereas eukaryotic cells, their energy is generated within the mitochondria. So that is something important to remember. Prokaryotic cells, energy is generated in the cell membrane. Eukaryotic cells, energy is generated in the mitochondria. The next structure, ribosomes. Both prokaryotic cells and eukaryotic cells have ribosomes. All cells have to have ribosomes because ribosomes are responsible for protein synthesis, and all cells have to have the ability to produce proteins in order to grow and divide. So even though both prokaryotic and eukaryotic cells can have ribosomes, there are some differences. Functionally, same function. They produce proteins. But structurally, there are some differences. So the ribosome, as we learned in the prokaryotic lesson, the ribosome is composed of a large subunit and a small subunit that come together during protein synthesis or translation to produce a protein. The size is different when we talk about prokaryotic cell ribosomes and eukaryotic ribosomes. With eukaryotic ribosomes, they're typically larger. Their large subunit is 60S, and their small subunit is 40S. And when the two come together, it totals an 80S size ribosome. Again, I know that doesn't make sense because it's not an additive property, but it's based on density. And then with prokaryotic cells or bacterial cells, their large subunit is 50S, their small subunit is 30S. So when those two come together during translation, it totals a 70S size ribosome. So eukaryotic ribosomes are bigger than prokaryotic ribosomes. So that's one difference. Another difference is location within the cell. So in eukaryotic cells, there will be ribosomes in the cytoplasm. There will be ribosomes within the mitochondria, and then there will also be ribosomes on the rough endoplasmic reticulum. In prokaryotic cells, the ribosomes are most often just dispersed in the cytoplasm. They obviously can't be in the mitochondria because bacterial cells don't have mitochondria, and they can't be on the rough ER because bacterial cells don't have rough ER. So they are typically just in the cytoplasm compared to those various other locations that eukaryotic cells would have them present. But functionally, ribosomes, regardless of cell type, still serve the purpose of maintaining, synthesizing proteins.